Biostatistics is the application of statistics to a wide range of topics in biology. The science of biostatistics encompasses the design of biological experiments, especially in medicine, pharmacy, agriculture and fishery, the collection, summarization, and analysis of data from those experiments, and the interpretation of, and inference from, the results. A major branch of this is medical biostatistics, which is exclusively concerned with medicine and health history. Biostatistical reasoning and modeling were of critical importance to the foundation theories of modern biology. In the early 1900s, after the rediscovery of Gregor Mendel's Mendelian inheritance work, the gaps in understanding between genetics and evolutionary Darwinism led to vigorous debate among biometricians, such as Walter Weldon and Carl Pearson, and Mendelians, such as Charles Davenport, William Bateson and Wilhelm Johansen. By the 1930s, statisticians and models built on statistical reasoning had helped to resolve these differences and to produce the neo-Darwinian modern evolutionary synthesis. The leading figures in the establishment of population genetics and this synthesis all relied on statistics and developed its use in biology. Ronald Fisher developed several basic statistical methods in support of his work studying the field experiments at Rothamsted Research, including in his 1930 book The Genetical Theory of Natural Selection. Sewell G. Wright developed statistics and methods of computing them. J.B. S. Haldane's book, The Causes of Evolution, re-established natural selection as the premier mechanism of evolution by explaining it in terms of the mathematical consequences of Mendelian genetics. These individuals and the work of other biostatisticians, mathematical biologists, and statistically inclined geneticists helped bring together evolutionary biology and genetics into a consistent, coherent whole that could begin to be quantitatively modeled. In parallel to this overall development, the pioneering work of Darcy Thompson in On Growth and Form also helped to add quantitative discipline to biological study. Despite the fundamental importance and frequent necessity of statistical reasoning, there may nonetheless have been a tendency among biologists to distrust or deprecate results which are not qualitatively apparent. One anecdote describes Thomas Hunt Morgan banning the Friedan calculator from his department at Caltech, saying, "Well, I am like a guy who is prospecting for gold along the banks of the Sacramento River in 1849. With a little intelligence, I can reach down and pick up big nuggets of gold." Almost all educational programs in biostatistics are at postgraduate level. They are most often found in schools of public health, affiliated with schools of medicine, forestry, or agriculture, or as a focus of application in departments of statistics. In the United States, where several universities have dedicated biostatistics departments, many other top-tier universities integrate biostatistics faculty into statistics or other departments, such as epidemiology. Thus, departments carrying the name biostatistics may exist under quite different structures. For instance, relatively new biostatistics departments have been founded with a focus on bioinformatics and computational biology, whereas older departments, typically affiliated with schools of public health, will have more traditional lines of research involving epidemiological studies and clinical trials as well as bioinformatics. In larger universities where both a statistics and a biostatistics department exist, the degree of integration between the two departments may range from the bare minimum to very close collaboration. In general, the difference between a statistics program and a biostatistics program is twofold. Statistics departments will often host theoretical, methodological research which are less common in biostatistics programs, and statistics departments have lines of research that may include biomedical applications but also other areas such as industry, business and economics, and biological areas other than medicine. Recent developments in modern biostatistics. The advent of modern computer technology and relatively cheap computing resources have enabled computer-intensive biostatistical methods like bootstrapping and resampling methods. Furthermore, new biomedical technologies like microarrays, next-generation sequencers and mass spectrometry generate enormous amounts of data that can only be analyzed with biostatistical methods. For example, a microarray can measure all the genes of the human genome simultaneously, but only a fraction of them will be differentially expressed in diseased versus non-diseased states. One might encounter the problem of multicollinearity. Due to high intercorrelation between the predictors, the information of one predictor might be contained in another one. It could be that only 5% of the predictors are responsible for 90% of the variability of the response. In such a case, one would apply the biostatistical technique of dimension reduction. Classical statistical techniques like linear or logistic regression and linear discriminant analysis do not work well for high-dimensional data. As a matter of fact, one can get quite high R2 values despite very low predictive power of the statistical model. These classical statistical techniques were developed for low-dimensional data. In cases of high dimensionality, one should always consider an independent validation test set and the corresponding residual sum of squares in R2 of the validation test set, not those of the training set. In recent times, random forests have gained popularity. This technique, invented by the statistician Leo Breiman, generates a lot of decision trees randomly and uses them for classification. Decision trees have of course the advantage that you can draw them and interpret them. Random forests have thus been used for clinical decision support systems. Gene set enrichment analysis is a new method for analyzing biological high-throughput experiments. With this method, one does not consider the perturbation of single genes but of whole gene sets. These gene sets might be known biochemical pathways or otherwise functionally related genes. The advantage of this approach is that it is more robust. It is more likely that a single gene is found to be falsely perturbed than it is that a whole pathway is falsely perturbed. Furthermore, one can integrate the accumulated knowledge about biochemical pathways using this approach. Applications of biostatistics. Public health, including epidemiology, health services research, nutrition, environmental health and healthcare policy and management. Design and analysis of clinical trials in medicine. Assessment of severity state of a patient with prognosis of outcome of a disease. Population genetics, and statistical genetics in order to link variation in genotype with a variation in phenotype. This has been used in agriculture to improve crops and farm animals. In biomedical research, this work can assist in finding candidates for gene alleles that can cause or influence predisposition to disease in human genetics. Analysis of genomics data, for example from microarray or proteomics experiments, often concerning diseases or disease stages. Ecology, ecological forecasting, biological sequence analysis, systems biology for gene network inference or pathways analysis.